Hello, my name is Julia Sumis, and I am the assistant editor at Global Food Safety Resource. And today we've been chatting with Dr. Matthew Moore, who is an assistant professor of food science at the University of Massachusetts Amherst. We had a very interesting conversation covering a lot of the research that your lab has been doing. And I wanted to ask one more question, if you don't mind. So foodborne viruses, such as enteric eukaryotic viruses, for example, are very life-threatening, and they clearly impact a huge portion of the global population. So would you mind briefly explaining the research that your lab is conducting on these viruses and how this work might be able to help food businesses understand some of the threats that they're facing?
Yeah, well, thank you for the opportunity. So our lab kind of does quite a diverse sort of set of projects, but you could really group them into sort of 3 or 4 main categories. So the first being, we do a little bit of work trying to study and understand the biology of viruses, in particular human noroviruses, as there has been some evidence, that the viruses interact with bacteria in our guts, and that in some cases the bacteria in our guts can potentially promote infection of these viruses. But in terms of more the food safety aspects of things, you could say we're really interested in the concentration and detection of these viruses. So they present a very unique challenge in terms of being able to detect them. And even upstream of that, being able to specifically concentrate these very, very small viruses out of a large complex food sample. This is something that's not very easy, and it's actually traditionally been sort of one of the primary hurdles to really coming up with cheap, affordable detection regimes for these viruses, because essentially, unlike bacteria, you can't necessarily have an enrichment or a growth step. You have to very specifically try and pick usually a very small number of these viruses out of a sample a food sample or environmental sample that may have a whole bunch of other molecules or compounds in it that can potentially inhibit your downstream detection technique. In this case, usually it's RT-qPCR.
Right.
And so our lab has a number of projects sort of related to trying to specifically pull these viruses out of a food sample and purify and remove all that other junk that's in the sample so that you can do a more effective downstream detection technique. And then our lab also does a lot of work on those downstream detection techniques, including a nanopore sensing based technology that may actually enable not only portable rapid detection of the virus, but also subtyping. So the type of sensing that we do here is nanopore sensing. It's not necessarily sequencing. So most people, when they hear nanopore, will think of nanopore sequencing, which is a portable sequencing technology that's also really promising. In this case, we use a slightly different nanopore. And this nanopore has a number of characteristics that actually potentially enable the binding event of whatever you're targeting to form a very unique fingerprint. And so our hope is that, in being able to portably detect these viruses, because you have a very unique footprint, you can actually subtype different strains of the virus. So if you can identify different strains, it makes it a lot easier to more quickly identify outbreaks. And then one of the other aspects of our lab, getting outside of the concentration and detection space, is trying to come up with and understand better ways of reformulating disinfectants to more effectively inactivate these viruses. So these viruses traditionally have been very difficult to inactivate with a lot of more commonly used disinfectants. They still kill some of the viruses, but they don't necessarily inactivate as many as you would like in theory. And so one of the things that we're trying to understand is how to come up with or restructure formulations so that they can more effectively deliver whatever your inactivation agent is to inactivate these viruses. We're also doing a little bit of fundamental work trying to relate and understand how the structure of these viruses enables it to be resistant to begin with these inactivation agents, as this isn't necessarily a topic that's been widely explored. And it's something that we're also really interested in because, from a more structural biochemistry standpoint, it's actually really interesting. I probably should have mentioned this at the beginning, but one of the remarkable things about the noroviruses, at least, is that their viral capsid is actually made up of essentially one major capsid protein, and it's about 180 copies of it that self-assembles into this sort of soccer ball shaped capsid that encapsulates the viral RNA. And even though that sounds pretty simple, they're really, really tough. So they've evolved essentially to survive your stomach acid, survive the proteolytic enzymes in your intestines, that you normally use to digest food. And they're also pretty resistant to a lot of inactivation agents. So one of the things we're interested in trying to understand is what aspects of the structure of that capsid protein enable it to be more resistant to these inactivation agents, with the idea that eventually we might be able to come up with some sort of predictive means of being able to predict based on the sequence of the viral capsid protein how resistant it is relative to other viruses.
Wow, this is some extremely exciting research. I am so excited to see what you end up finding at your lab and how this will alter the industry itself. I think this is really important work. And thank you so much again for sharing all of your research and your findings with us. This has been a very fascinating interview.